Welcome to Ace My Exams Learning. Let us get started with today's learning.
What are viruses? Viruses are extremely tiny infectious agents that are much smaller than bacteria. Due to their microscopic size, they cannot be seen with a normal light microscope and require a more powerful electron microscope for observation. Unlike bacteria and other microorganisms, viruses are unique in their structure and function, which makes them different from all known living organisms. Structure of viruses. One of the key characteristics of viruses is that they are acellular, meaning they do not have a cellular structure. Unlike bacteria, which have cell walls, cytoplasm, and various organelles, viruses lack these components entirely. They do not possess a nucleus, which in most living organisms contains genetic material, nor do they have cytoplasm, which is essential for carrying out cellular functions. Additionally, they lack organelles such as mitochondria, ribosomes, and endoplasmic reticulum, which are required for energy production and protein synthesis. A virus is made up of genetic material, which can be either DNA or RNA, but never both. This genetic material carries the instructions needed for the virus to reproduce once inside a host cell. Surrounding the genetic material is a protective protein coat called a capsid, which provides structural support and protection. Some viruses also have an additional outer layer called an envelope, which helps them infect host cells more easily. The envelope is usually made up of lipids derived from the host cell's membrane, along with viral proteins that allow the virus to attach to and enter new host cells. What is a bacteriophage? A bacteriophage, or simply a phage, is a type of virus that infects bacteria. Unlike viruses that infect humans or animals, bacteriophages are specialized to target and infect bacterial cells. These viruses are fascinating because of their ability to specifically recognize bacteria and utilize the bacterium's cellular machinery to replicate themselves. The infection process typically results in the destruction of the bacterial cell, releasing new bacteriophages that can then go on to infect other bacteria. Bacteriophages are highly efficient in their design and function. Their structure has evolved to specifically infect bacteria by latching onto the bacterial surface, injecting their genetic material, DNA, and taking control of the bacterial processes. Once inside, the virus forces the bacterium to produce more viruses, eventually leading to the bursting of the bacterial cell, a process known as lysis, which releases new viruses. Components of the bacteriophage structure. The structure of a bacteriophage is specifically adapted to ensure successful infection of bacterial cells. Each part of the bacteriophage plays a key role in the process of infection and replication. Let's break down the major components. 1. Protein shield, capsid. The protein shield, also known as the capsid, is the outer protective covering of the bacteriophage. This structure is made up of protein molecules that fit together in a precise, structured arrangement. The capsid serves two main functions. A. Protection. It protects the viral DNA from damage by environmental factors such as UV radiation, enzymes, and changes in temperature. Without the capsid, the viral genetic material could easily be destroyed before it can infect a host. B. Delivery of DNA. The capsid also ensures the safe delivery of the bacteriophage's genetic material into the host bacterium. Once the virus attaches to the surface of a bacterium, the capsid opens, allowing the viral DNA to be injected into the bacterial cell. 2. DNA, genetic material. The DNA of a bacteriophage contains the genetic instructions needed to hijack the bacterial machinery and reproduce new virus particles. The DNA is stored inside the capsid until the bacteriophage attaches to a bacterial cell. Once attached, the bacteriophage injects its DNA into the bacterium, which then begins using the bacterial resources to replicate the viral DNA and assemble new bacteriophages. The bacterium eventually bursts, releasing these new viruses to infect other bacteria. 3. Collar. The collar is a ring-like structure located between the capsid and the tail of the bacteriophage. This component provides stability by securely connecting the capsid to the tail. The collar's role is important because it helps maintain the integrity of the viral structure during the infection process. Additionally, in some bacteriophages, the collar can help guide the DNA from the capsid through the tail and into the bacterial cell, ensuring that the infection process happens smoothly and efficiently. 4. Base plate. The base plate is located at the end of the bacteriophage's tail and is an essential part of the infection process. It acts like a docking station, allowing the virus to securely attach to the surface of the bacterial cell. Once the bacteriophage is attached, the baseplate undergoes structural changes that facilitate the injection of the viral DNA into the host cell. The baseplate is crucial in triggering the release of the viral DNA, enabling the bacteriophage to begin its replication cycle inside the bacterium. 5. Fibers. The fibers are long, thread-like extensions that project from the baseplate. These tail fibers play a critical role in helping the bacteriophage identify and attach to the correct bacterial host. They act as sensors, searching for specific receptors on the surface of the bacterium. Once the fibers find and attach to these receptors, the bacteriophage can proceed with the infection process. The fibers ensure that the bacteriophage infects the right type of bacterium, which is crucial for the success of the viral replication cycle.
To access more learning and exam preparation materials, go to www.acemyexams.coza. This link is also in the video description below. Thank you for watching this video. Remember to subscribe and be the first to know when we upload new videos.